of all, I'd like to thank Lynn and all of you for being here. And it's a great pleasure to see smiling young faces. So those of us who live at universities, stay at universities because of the smiling young faces. And as Lynn said, that I've been doing research. She didn't exactly own up, but I'll say I got my PhD in 1974, and so I've been a practicing neuroscientist since about 1970 or 1971. And so I have, one of the things I have is sort of the vantage point of time and watching the field develop. The first Society of Neuroscience meeting I went to had 500 people in 1973, and now the meeting has 30,000 people. And so the field has grown as the excitement about the field of neuroscience has grown, and as our ability to do things to really understand how the brain works grows. It was a tremendous challenge and fascinating process to be part of the original working group for the Brain Initiative. And I'd first like to, oh dear, where did that come from? I'd first like to sort of go through a little bit of sort of introductory slides to give you a sense of where we were in the field when the Brain Initiative was first starting, and then to tell you why I think it's been such a revelatory experience to watch it. So before I start, I should say those colorful things aren't just things. They're individual neurons that have been filled with fluorescent dyes. So what we do is we take a microelectrode, a glass electrode, we fill it with dye, and we put it into the cell body, or those big round things, and we inject dye, and then the dye travels throughout the whole cell, and you can then see the structure of the cell. And they're exceptionally beautiful, and they're even more beautiful when you see them under the microscope. But one of the things I'll tell you in a moment, I've worked on the small nervous system my entire career, and the reason why we started working on these small nervous systems is because it was possible to really think about going from single neurons to circuits to try and explain the way a circuit worked on the basis of the interactions between individual neurons. And when I started, it was only possible to even think about doing that in a small nervous system, such as the one that I study. And the goal of the Brain Initiative was to take that same way of thinking, or that same approach, and try and understand large circuits, such as are in your brain, and really try and account for how large circuit dynamics, how your brain is actually doing a task, listening to me, I hope, while you're eating. And yes, you probably can do those two things at the same time. But to really try and be able to do for those large circuits what we can, up to that point, have only been able to do with small circuits. And in order to get there, we had to go through a process of a major amount of technology development. And that's what makes the brain so, not the Brain Initiative so incredibly wonderful. So this was the challenge always, to go from neurons and synapses to behavior. And neurons are complicated enough by themselves, but then when you start putting them together, you end up with what we call emergent processes that are not that simple to understand. Now, just to give you a little bit of sense where we were and are in some ways, the small nervous system that I work on is the crustacean stomatogastric nervous system. It comes from lobsters and crabs, and those of you who come from Maine will appreciate that. And these recordings, these are electrophysiological recordings that I just want you to see, to have an idea about how we actually think about trying to get a sense of what groups of neurons do together. So in that top panel, you see electrical recordings that have been made from individual neurons during a rhythmic motor behavior. In this case, it's a lobster crab chewing and eating its food, but in your case, this would be a lot like what's going on in your respiratory centers or in your spinal cord as you're walking, etc. So they are these beautiful brain rhythms, and one of the reasons why I was always studying this nervous system is because the output of the circuit is so easily measurable. And one of the things that we were looking to do in brain was to come up with the tools that would allow us to measure the output of more complicated brain circuits with the kind of faithfulness and reliability that we can do in small circuits. On the bottom, you see what we called a connectivity diagram. Now it's called a connectome. And what you see in each of those circles is a two-letter name of a neuron. And this is a wiring diagram. It tells you who's connected to who. Now, the ganglion I work on has 30 neurons, and it wasn't that difficult to come up with a wiring diagram. But if you think about a circuit in your brain with millions, hundreds of thousands or millions or many millions of neurons, what you also need is a connectivity diagram or a wiring diagram that can allow you to understand how a circuit works. And so 15 years ago, these kinds of recordings and this kind of wiring diagram were only possible in small circuits. Now, one of the things that was necessary to be able to do here was to identify the neurons, to know which neuron was which. And that is a major challenge for the first part of the brain initiative, was to come up with methods that will allow us to identify the types of neurons all over your brain, because you can't think of coming up with a wiring diagram unless you can identify the constituent neurons. Does that make sense? So if any of you have read through, or if Walter shows you, one of the major initiatives was to really just try and identify neurons, find out how many cell types they were and what they were like. Okay, so I think, now I'd just like to say one more word about me, because whenever you have an audience, it's all about you, right? And that is the problem that really, really, really interests me today is, and I study this still with lobsters and crabs, but it's what I'll call differential resilience of circuits with very similar performance. I can have 3 crabs that are producing what looks like the same motor patterns, and they will nonetheless be differentially resilient to environmental challenge. And we're really trying to understand this at a mechanistic level. And this is incredibly important as we think about real environmental challenges like climate change, and then the challenges that come with disease. And one of the things that we see is you can have a whole host of individuals that look the same when they're normal and healthy, but they respond to challenge, all kinds of challenges, and completely differently. And this, I think, is the major, major, major challenge for trying to understand disease, because understanding how the individual differences in wiring diagrams or molecules, how they're hidden or they're cryptic in health, but yet they might mean that person X can take drug Y, but person Z can't. And I think one of the things we have to do is try and really understand on a basic level these sorts of issues so that we can then think very creatively about disease. Now, brain happened at a remarkable time in neuroscience. There had been, in about the 10 years before brain, the development of sort of new technologies that were transforming the way we were thinking about being able to do things as we tried to understand large circuits. And so when they asked us to brainstorm about what additional money could do to facilitate us getting real understandings about how the large circuits in your brains work, we were sitting there with the beginnings of some new technologies, but these new technologies were only in their nascent forms. And so one of the real things that we wanted to do was to capitalize on the momentum that was there in the field and then really see if we could, really make a transformative change. Now, the issues that we discussed at the beginning of the brain, we argued about. We argued very nicely, but we argued. And that was, among things you'll know about, the balance between larger group projects and smaller investigated and initiated projects, the importance of new technology development, model systems like lobsters and crabs or flies or worms or mice, and human studies, how we would go about bringing investigators from all avenues of science into studies of the brain, and then how to confront the role of big data. Big data is now on everybody's, on sort of in everybody's mind, but it was at the beginning where it was clear that big data was going to be really important. And then how do we deal with training and ethics? So those were the issues that we were really confronting. And we decided after a lot of arguing, we didn't really argue. Yeah, we argued. But we argued with great respect. I mean, this was a fabulous, that first working group was a fabulous group of people. That recording and manipulating cells to understand circuit function would be catalytic. And that was, there was a lot of work on the human nervous system that wasn't really grounded in biology. And then there was a lot of very basic work, but there was this big chasm in the middle, which is how large circuits worked. So among the things that we suggested doing, and Walter will have a more complete list, was talk about cell identification and cell types, because you can't think about trying to understand these large circuits without that. And the connectivity, thinking about large electrode arrays so you could record from many neurons simultaneously, recording one by one, which is what we had been doing in the field, only gives you a tiny, tiny, tiny bit of insight, whereas you get much more insight with larger numbers of neurons. New optical methods to record and stimulate, algorithmic improvements and data analysis for recordings and behavior, and taking advantage of new opportunities to obtain data from humans. I think this is a really fabulous thing going forward, creating new teams of investigators, and fostering the improved quantitative and computational skills of brain scientists. So people ask me, the Brain Initiative, was this grand challenge living up to its expectations? From the trenches, I'd say an incredible resounding yes. 3 years, 4 years out, when it was that, I was just stunned. I never imagined when we set out this blueprint that it would go as fast as it went. And I'd like to say that I think the reason it went as fast as it did was because there was an excitement in the field that was engendered, and the NIH, with their help from NSF and other places, were incredibly good stewards. I don't know that you probably have people tell you this so often, but the people who work in grants, running the programs at the NIH are truly wise and dedicated, hardworking people. And I think a lot of the success of the rollout of the Brain Initiative was the way in which they managed the money in the programs. And so I've just been stunned at how extraordinary the progress has been, and Walter will tell you about that. And I don't think I ever would have dreamt we would be here where we are today 10 years ago. Now, I'd like to just end on a little bit of a concern, and since I don't often have congressional staffers to talk to, I'd just like to say, I think this is absolutely, for science in general and brain science in particular, the best of times, in some ways the worst of times. It's the best of times because there are things we can dream about doing that were inconceivable 10 years ago. And we have the starting ability to start thinking about things, and it's just incredibly exciting. The thing that concerns me, which is where the worst of the times comes from, is that for a variety of reasons, including the sort of plateau in NIH funding and NSF funding that we went through for a long period of time, there's a level of anxiety about the future that young scientists have. And they're worried, and now it's even worse, they're worried about not only their future as scientists, but their future of the planet, et cetera. So they're just very anxious. And one of my challenges as a senior scientist is to keep this incredibly enthusiastic, optimistic, saying, yes, there is really an exciting future in doing science. And I would just like to say that anything that can be done to maintain or create to foster a sense of, there really is an incredibly exciting future in doing science is really, really, really important, because we've invested a lot of time and energy in creating this cadre of exceptionally well-trained young people who are very, very frightened that the rug is going to be pulled out of from under them as they've watched funding levels go up and down and there not being enough jobs, et cetera. So to my mind, I sit there and I say, this is the best time in the world to ever think about going into science. And they feel that and they want to do that, and then they're worried. And so anything you can do to make them less worried would be great. And I'll stop there.
So I think Eve really hit the nail on the head and put out the most important parts of the Brain Initiative, which is really trying to understand circuits of the brain. And so when you think about it from the human side of it, which I'll stress more in this talk, the brain is what allows us to do what we can do: talking, sleeping, running, thinking. And in our society, being able to pull a plow is not necessarily going to guarantee success. It's all related to how well your brain can contribute to society. And so the brain, is incredibly important. As we get older, we face these issues of the aging brain, but we also have tremendous disability due to developmental disorders. And these brain disorders span what we call psychiatry, neurology, and substance abuse disorders. So people say it's in your head. Well, everything's in your head. There is nothing that's that you experience that's not going through your brain circuits. But as Eve said, we had really no way of really understanding these circuits. They were really a black box. I mean, she showed you the kind of work she could do in a small circuit, and people did that in non-human primates, and they did make some discoveries. But it's very, very hard when you can just see one cell at a time, record from it, and you know that there's a million cells firing in a certain pattern, but you don't know what that pattern is. And so amazing that anything worked, but things did work, like deep brain stimulation for Parkinson's is an example where people use those technologies in non-human primate brains and figured out that, well, if you stimulate in this region, you might make the symptoms of Parkinson's go away. But it's amazing that it's as effective as it is. But it's that carrot out there that's pushing everybody to understand circuits better so that we can manipulate them for health. And then if you think about people who have brain disorders, whatever the brain disorder is, the thing that affects the person is the disordered circuit. So you could have a stroke or you could have Alzheimer's, but how that affects your life depends on how the circuits have been disturbed. And so if you want to fix them, you really have to be able to monitor them and modulate them, and that's what we have never been able to do because we didn't have those technologies to see brain circuits in action. So that's what the Brain Initiative is all about, and I can just give you an example of technology. So when I came out of medical school in '75, that picture on the left was one of the first CT scans and of a brain, and we were really excited to be able to see basically nothing. There's like no definition except the black stuff in the middle, which is the water. But look what happened in the ensuing years. Now we have MRI scans that basically look pretty much like you would get a histological slice. So technology in terms of the structure of the brain, and even in the human side, really changed over my career. But that doesn't tell you anything about what's going on in the brain. There's no measure of the circuit activity, and that's the key thing about the Brain Initiative. Now, the circuits, as Eve said, are complicated, and they're so complicated that most of us believe that there is a language that the brain is communicating with inside in its circuits, but we have no idea what the words are or what the paragraphs are or what the chapters are or what the book looks like, because we haven't been able to do anything except kind of measure those squiggles she showed you, which are the action potentials. So the letters we know. The letters are when a nerve fires, but how that creates the symphony of our thought allows us to write a paragraph. That fundamental process by which the brain does that is completely opaque to us. And the complication is basically a scale problem. So Eve talked to you about a circuit of 30 cells. Your brain has 85 billion neurons, equal number of glia, and trillions of connections. So the wiring diagram is unfathomable to try to put together until recently. And the Brain Initiative has got people to think that we could put the wiring diagram, and they have done it, the wiring diagram for, say, a Drosophila fly. Maybe a mouse is going to be doable at the scale of every single connection. And the human, probably that seems inconceivable, but you never know. But there are now other ways to look at the maps of the human brain where you can get these kind of connections. And this technology that I just showed you in this movie is a clearing technique where someone figured out how to take the lipid out of the brain so it became transparent. And now you can use any molecular tag you want, and you can see its 3D distribution. And you can do that on human brain as well as this is a mouse brain. So a technology, as Eve said, was percolating along, and then bang, it just hit, and it's been incredibly improved in just the last couple of years. So the Brain Initiative has these 7 pillars to it, which I'll talk to you about. But the thing to remember is it's all focused around the 3 M's: mapping, monitoring circuits, monitoring circuits, and modulating circuits. Those are the 3 M's that we're looking for. And we do this in the circuits that are best amenable to understanding these basic processes of the brain. And then we also jump. There's a lot of gaps in our knowledge, but we're hopeful that even with the low-level knowledge we have, that we can potentially do that in humans to help for health. And sometimes we fail, and we fail a lot, and we fail because we don't have those gaps are way too big. It's like trying to cross a river. You can see the stones, but you don't know how far apart they are until you get to the one that I'm not going to make it to that next one. And so that's oftentimes how we learn. So brain cell types, Eve told you about the importance of understanding all the different types of cells, and then the circuit diagrams, how you put them together, and then the technology to monitor the activity. So you're not just looking at an action potential firing, but you're looking at 1 million cells at a time firing. And then the ability to precisely manipulate those circuits and those cells. And amazingly, technology has developed to allow us to do all those things over the last 5 to 7 years of the Brain Initiative, to the point where things have happened that no one expected could ever be done. So just an example, someone figured out a holographic optical technique where you can stimulate multiple cells simultaneously by light, and steering the light to just do that precisely. Something that I don't think anybody could imagine you could do that 5 years ago. But now it's being done routinely in some of these high-level labs. And then the area that Eve's been very prominent in is taking this information and trying to understand the fundamental principles. What is the language of the brain? How do the brain cells communicate with each other and process information to give the result, the behavior? And that's the theory and data analysis. And then the goal at the end is for NIH. We're focused on understanding the science and turning that into advantages for health, and integrating all of these kind of technologies. So the Brain Initiative, very wisely, was started out at a small scale, and then Congress had the wisdom, I don't know where they got this, but it was a great idea, to slowly ramp up over time the expenditures so that allowed NIH to build something that was solid and working, and then build on top of that, and then build on top of that. And that's been really the secret to the success. But the Brain Initiative, actually, we're looking at the second half coming up, and more funding is in the second half than the first half. So whatever I tell you now, we're hoping that it's going to be multiplied by 10 in terms of results by the end of this. And we're not and so I'm talking here from NIH, but we're not the only agency involved in the BRAIN Initiative. So National Science Foundation, incredibly important part of the BRAIN Initiative because, for instance, to understand the language of the brain, it's a mathematical answer. It's going to be it's going to be a computational person who gets in and starts to understand this stuff. So people who are coming from fields that don't know anything about biology are incredibly important to us now, particularly on this informatics side. But if you're building something like steering light, that's also not a biologist. That's an optical physicist who becomes interested in neuroscience. And we've been lucky to get those kind of people because the focus was on building tools, understanding theory, but we need to do that even more and more and more to advance the BRAIN Initiative because in the beginning, the problem was we couldn't get the data. The problem now is we can get the data, but I don't know how we're going to figure out what the data means. So the acuity of that need is growing tremendously. And then private groups, the Allen Institute, the Kavli Foundation, the Simons Foundation, IEEE, the Engineering Society, have been really important partners as we march along with the National Science Foundation I mentioned, IARPA, and the FDA, because some of the things we're producing go through the FDA, go into people. Now this will work, hopefully. Okay. So this is an example of what we can do now that we couldn't do when we started. And so as Eve said, she used to put a little electrode in a cell and measure the recording of that cell. I don't know, how many cells did you get up to in a prep? 4 or 5? Yeah, so 4 inches cellularly. I used to do 2 at a time. I thought that was good. But here, these techniques from the BRAIN Initiative, genes into cells that make the cells light up. So you don't have to put an electrode in. You just have to have a camera to see the cells when they fire, because when they fire, this optical technique lights up. And this kind of work came from studies in the most primitive organisms who have these proteins that are affected by light. And finding the gene, putting it into the neurons, now you see a cell light up. But now you can see in this slide here, this is a blow up of the mouse brain surface, and there might be, say, 150 neurons that you're seeing in the panel, but the entire brain is lighting up. And you can do this in a behaving mouse. So you can have a mouse, and as they eat, you can see what is the circuit doing, and you have this amazing amount of data now at your hand, on your lab table. Now, going to humans, so there's a lot of spaces in terms of the understanding because of these dramatic scales where information is being processed at a very tiny scale, a nanomolar scale, where molecules interacting or 2 neurons are interacting, to multiple neurons interacting like you saw in Eve's, to multiple different nuclei of neurons all over the brain interacting. And the scale is what is scary. But there are ways of trying to use what we have to make some progress in the human space as well. So this is a work that's done in humans, and it's people who are undergoing monitoring for epilepsy surgery. And they join the research, and while they have electrodes over their brain to look for the area of seizure, you can record from those parts of the brain that are involved in speech. So as Eve was talking about, the output there was the motor output to the gastric muscles. Here it's the motor output to your larynx. And what they've been able to do is record well enough so that you can actually so someone can actually think about what they want to say, and that activity goes to those muscles, and you can record that activity, and you can predict what they're saying even without them saying it. So so in terms of monitoring brain activity, it's a great example of how you can take that activity and understand what behavior you're getting from that activity. Okay. And this one is even more amazing. So this is not published, but it's a Brain Initiative grant that UCLA put out a press release. And what it is a device that is in the glasses, and this is a person who's blind because their eyes were damaged in an auto accident. And the input, the visual input going into the video glasses is then sent and put right into the visual part of the brain and stimulate the brain. And what this person has been able to do, so they don't actually see like we see, but before this they saw nothing. Now they see bright lights when someone comes in the room, when they're crossing the street and a car is coming, when there's numerous books on the table, they can actually find the books. This guy in this says that when his wife's mad at him, she doesn't talk and she's quiet, so he doesn't know where she is. But now he can find her. But amazing example, so this is like the first step, but the hope here is that we can actually restore vision by going straight from a camera right into someone's brain. And there are other examples called brain-computer interfaces with people who are paralyzed, where we take the activity out of the brain and we control a computer or a prosthetic device. So those things are actually happening in small numbers of people who are in particularly tough situations. Now, as you can imagine from what I told you about the monitoring brain activity or manipulating brain activity, here we restored some aspects of vision to someone, and then I showed you how we could basically read out the electrical activity to know what someone wanted to say. So you can think that not too far in the future, people are going to think about doing things that maybe we don't want them to do as a society. So that gets you to these neuroethics or the Brain Initiative. So we have been cognizant of this from the very beginning, and actually when the president declared the Brain Initiative, he also had his Bioethics Commission basically examine the Brain Initiative for what are the neuroethical issues that could potentially come up. And they are coming up, sometimes in unpredictable ways. But we have had a group right from the beginning that works with the ethics, with our investigators, with NIH right from the get-go. And so they have, for instance, put out a guiding principles of neuroethics to steer us when we decide on grants, or steer our investigators when they start to run into tricky issues. And they can do consults and help kind of negotiate those kind of issues as they come up. But the truth of the matter is, society is going to have to make some choices at some point about how these technologies get used outside of health. I think in the health space, we have a lot of experience in doing this, but if you think about circuits, and so Eve talked to you about resilience, how does a perturbation affect the circuit? Well, what we do every day is we send our kids to school, and what we do in school is we train their circuits. So you're not born with a circuit to do math, but you leave school with a circuit to do math. So education is probably the most common way in which we alter circuits for a good purpose. And we don't know, no one knew, no one had the opportunity to look and see how are those circuits being changed. That could change. And we could find out ways in which we can stimulate the brain to make those circuits work better and learn faster, for instance. Those I don't think, are inconceivable. But you can also think of some more malign things that people could get into. So that's something I think that society is going to have to grapple with at some point in time. And so we've been talking about it. We have these groups, and we've trying to get those messages out, and certainly very careful about what we do. So, so I think just to sum up, through 2018, the Brain Initiative has invested nearly $1 billion, and we're so grateful for the support from Congress. It's supporting 500 research projects, hundreds of investigators, and I think pretty much everyone agrees that the expectations have been exceeded, and strange, amazing things have happened that no one knew. But we think that the brain, the second half, is going to be even more exciting, so stay tuned. Some of the things that I told you about impacting health, I mentioned some of these stimulation devices. We also have stimulation devices that are now better than the ones we had before for Parkinson's and essential tremor. We're trying to find areas to stimulate in people who have depression, obsessive compulsive disorder, and even addiction and chronic pain. And so we're really looking forward to the next half. And then, as Eve said, the key to the Brain Initiative is the future, what's coming down the road, and that we put a lot of emphasis in training and bringing people into this work, so that the fruits of the investment are realized in the ensuing decades. So with that, I think I want to end, and thank you very much for your attention, and we're happy to chat and answer questions.